Hello and welcome. In this video, I would like to talk about the Turtle Heart Demo Lab, which is lab number 8 for human physiology. And I'd like to point out some of the concepts we'll be testing in this lab and explain what you can do as a student to be successful in this lab, as it's transitioned from what used to be a demonstration in a laboratory to an explanation with some videos and data on the internet. So what you should be able to get from this video is we'll talk a little bit about the lab setup. So I'd like to explain what we would normally be doing in the laboratory. So we'd collect data from a turtle and its heart activity. I'll also take a few minutes to talk about how to complete this lab and how to read the data. So you guys will be using some unique software to read data that we've provided for you that you will then be analyzing to answer questions on page 111 in your lab manuals. So I'll show you what that data looks like and how to read it as we give it to you, and how to interpret it in terms of the physiology behind the data. Also, there are 6 experiments in today's lab. Experiment 1, we'll talk about the cardiac cycle, where we will be looking at the activity of a turtle heart on an ECG and describing the physiology. In experiment 2, we will be assessing the parasympathetic stimulation of the heart and describe what happens when the vagus nerve is electrically stimulated. In experiment 3, we'll be measuring Frank Starling's law of the heart, where we will describe the physiology between the amount of stretch and overlap in muscle fibers and contractile elements. In experiment 4, we'll be taking a look at the refractory period in a heart muscle contraction as we attempt to stimulate the heart during certain parts of the cardiac cycle. In experiment number 5, we will be stimulating the heart with electricity and checking to see whether it's undergone TREP summation or tetany, and then seeking to understand why or why not. And then in the last experiment, we will be measuring the autogenic property of the heart, where we'll be looking at a video of a heart that's been removed from an animal and describing the physiology of what we see in that heart. So let's start out and talk about the setup for this lab. So what we start with is a turtle, and we take that turtle and we want to measure its electrical activity of its heart. So we add some electrodes, creating, remember, Einthoven's triangle around the heart. We then hook those electrodes up to the BioPack unit, which gives us a display of an ECG or the PQRST complex on the computer. We are also going to take that heart, once we've exposed it, and we're going to attach a cord to it that is also attached to a force transducer. Remember, we use this in the muscle physiology lab, where it measures force generated by pulling. So when the heart contracts, when the ventricles of the heart contract, then we expect it to pull, and we'll be able to measure the amount of force, and that will look something like this on the computer. And we will be referencing this setup as it changes for the other experiments later. So now let's go ahead and take a look at what your data should look like for this lab activity. So for some of the experiments, we will be using a stimulator where we electrically stimulate, and that will show up on the top here. That will only be for experiments 2, 4, and 5. We will also be measuring the force transducer reader, or the amount of force that's exerted by the heart on the force transducer during ventricular muscle contraction, which is also a representation of stroke volume, or the amount of blood that's being pumped out of the heart during ventricular contraction. And we'll also be reading the ECG or electrocardiogram data that will be coming from the electrodes that are on the turtle as well. You'll also notice that there will be a journal opened in the bottom as you look at your sample data for this lab. So before we take a look at the data for experiment 1, let's take a look at the cardiac cycle and talk a little bit about what's going on here. So the cardiac cycle can be divided into 2 parts. Those are what we call diastole, and systole. So what's happening during diastole is the ventricles are relaxing and filling with blood, and what's happening during systole is the ventricles are contracting and ejecting blood. So remember, we're concerned about what's happening with the ventricles. They provide the blood that's moving through both the pulmonary and systemic circuits of the heart. And as you could guess, diastole takes a little bit longer, about 2/3 of the time, and systole takes a little bit less time. About 1/3 of the time in the cardiac cycle is spent in systole. And these two parts of the cardiac cycle continue because of what we call pacemaker cells in our heart. And typically those are found in the SA node. But let's talk a little bit about the difference between the two types of cells in the heart. Those include what we call the pacemaker cells and the contractile cells. So pacemaker cells utilize something called a pacemaker potential. And if we look at the action potential of pacemaker cells, it looks something like this. The first one's more accurate. Anyways, what's happening here is as the cell depolarizes, we have something that we call the pacemaker potential, and then it repolarizes where it will depolarize again spontaneously when those funny sodium channels open. And these pacemaker cells will continue to spontaneously depolarize and repolarize, setting the pace for our heart rate. In contrast, if we look at the action potential of the contractile cells of the heart, which would be all of the cells of the heart, not including those that can act as pacemakers, now their action potential looks more like this, where we have this rapid depolarization, and then we have this long plateau phase where the cell remains depolarized. And what's interesting about this is it actually overlaps with heart contraction, and that has some important implications on some of the concepts that we'll look at in the later experiments. Now, can you see that says contraction? Beautiful. So if we're looking at experiment 1, you should notice that in normal conditions, as we start to look at the activity of the turtle heart on the ECG, we should see this P, QRS, and then T wave showing up on the ECG reading. But also you'll notice as this occurs, we can see the tension that's exerted on the force transducer by the heart as it contracts, or as the atria contract, we'll sort of see this little blip here, and then we see the force go up significantly as the ventricles enter systole, and then we'll see that force decline as the ventricles relax into diastole, which should lead right up into the next systole. And so we should also notice that the atria contract preceding ventricular systole, which is what happens when the atria contract. They send blood into the ventricles, and then the ventricles contract right afterwards. So we should be able to notice that on the diagram. And if we are comparing the ECG wave to the force transducer data, we'll notice that the P wave that occurs right before atrial systole, and the R wave occurs right before ventricular systole. And this makes sense if you'll remember the P wave represents the depolarization of the atria, which should happen right before the atria contract, and then the R wave, or the peak of the depolarization of the ventricles, happens right before the ventricles contract. So for the next experiment, what we will be doing is we will be measuring the effect of the parasympathetic nervous system on the heart. And what we'll be doing is we'll take our typical setup that we had from the first experiment, except now what we'll be doing is exposing the vagus nerve in the turtle, and then we will take an electrical probe and we will stimulate or shock that nerve, and then that will significantly increase the activity of the vagus nerve. which synapses with the SA node, and that should release acetylcholine on the SA node and on the heart. And as that happens, we will be looking at the ECG reading and taking a look at what the effect on the ventricular contraction is during that time. So what effect does the parasympathetic nervous system have on the heart? Well, it mainly works to decrease the heart rate, and the vagus nerve is the main actor there. And what it does is it releases acetylcholine on the SA node, which increases the number of sodium channels opening, and it causes the cells to hyperpolarize, thus lengthening the time to threshold. So if we looked at the action potential for those cells, we may see a much longer time to threshold than we typically would, and that would result in a slower heart rate. In contrast, the sympathetic nervous system increases the heart rate via the sympathetic nerve cord. It utilizes norepinephrine or epinephrine, and basically what it does is it increases the rate of sodium channel and calcium channels opening, causing the cells to depolarize faster, so we may see a steeper depolarization of the action potential for those cells. So one thing that you may notice during this activity on the data is the amount of acetylcholine that's released may slow the heart down to completely stop beating, which we see here. But because the heart will be relaxed for such a long period of time, the first heartbeat or the first ventricular contraction following that pause might be a little bit taller than those previously, and that mostly just has to do with the fact that it's been filling longer, and there's then more stretch on the heart cells and it gets a stronger contraction, which leads us into our third experiment. So for experiment number 3, we will be testing the Frank Starling's law of the heart concept. Remember, this is basically what goes into the heart must come out. So if more goes in, then more will come out, and the heart will have to compensate. So we see stronger contractions when the heart fills more. So we saw that in the previous exercise. The way that we will be doing this in this setup is, remember, we have a cord attached from the heart to the force transducer, and we will be stretching the heart, which will simulate the increased end diastolic volume. So basically what we will do is we will attach something to the heart, and we will add tension to that, and then when the heart, when the ventricles contract, once they've been stretched, we expect to see a response, or we expect to see a stronger contraction when stretched to the optimal length. And then we expect that contraction to get weaker if we exceed the optimal overlap. So what do I mean by optimal? Well, if you remember, the Frank Starling law of the heart has to do with the length-tension relationship between muscles and muscle cells, and because the heart is composed of muscle cells, it still has to abide by this length-tension relationship. And so basically what we know about this is, if you look at this graph over here, if a muscle cell is understretched, then the amount of force it can, or tension it can exert, is low. There's an optimal amount of overlap between the actin and myosin, and if the muscle is overstretched, then there is less overlap between the actin and myosin and less force that can be exerted. So there's an optimal area where the maximum amount of tension can be exerted by a muscle. Well, heart cells are the same way, and when we stretch them to a certain point, we expect the tension to increase. And if we stretch them past that point, we expect that tension that they can exert to go back down. So if you look at the data down here, you'll notice we may see the tension on the heart increase as we stretch it. So for experiment 4, what we will be doing is we'll be taking a look at something called a refractory period of cardiac muscle cells. And what we'll be doing is we'll have our normal setup. But in